Imagine for a second that you've been tasked with building a massive state-of-the-art supercomputer.
Oh, that sounds expensive right off the bat.
Right. But there is a catch, a ridiculously huge one. You have to wire the entire power supply, laying down, millions of microscopic cables and power nodes while the computer's already turned on.
Wow. Okay.
Yeah, not only is it running, but it's actively learning how to process data. And the motherboard itself is physically expanding in size while you're working on it.
That sounds like an absolute engineering nightmare.
Exactly. You absolutely cannot power it down to install the hardware. One dropped connection and the whole system crashes.
Which is, impossible for a human engineer to pull off.
Totally impossible.
Yeah.
But that is exactly what the mammalian brain has to do right after birth. And welcome to today's deep dive, where we're going to get into exactly how that happens.
It really is a logistical miracle when you start to think about it. I mean, the brain's power supply is its vascular network, right? The miles and miles of blood vessels. Oh. And at birth, that network is incredibly rudimentary. It's barely a sketch of what it needs to be. It has to rapidly grow to support the brain's massive, compounding, computing power all without interrupting the flow of life for even a single second.
Okay, let's unpack this. Because today, we're looking at a groundbreaking, honestly, incredibly detailed research paper from the journal Cell.
Yeah, it's a massive team of researchers led by Elisa Delanois, Sophie Scribine, Etienne Dumazan, and others.
Right, and they've essentially mapped out the entire hidden choreography of how the mouse brain develops its vascular network after birth.
And our mission for this deep dive is to explore a brand new, powerful mapping tool they created. It's called LAMBADA.
Which is an amazing acronym.
It really is. And we're going to break down the 3 distinct, highly coordinated phases of how the brain wires its blood supply to match its computing power.
And most importantly, we're going to explore why understanding this delicate neurovascular dance, fundamentally changes how you understand the physical infrastructure of your own mind. But before we can even begin to understand how the blood vessels grow, we have to talk about the sheer technological hurdle of just trying to look at them. I mean, the brain is completely opaque.
Yeah, it's just dense, squishy tissue.
Right. How do you map microscopic, hair-thin blood vessels in 3D across an entire intact brain?
Well, historically, you couldn't, at least not comprehensively. The old way meant you had to physically slice the brain into, thousands of paper-thin sections.
Oh, that sounds tedious.
Incredibly tedious. You look at them individually under a microscope and then try to digitally stitch them back together. It was prone to massive errors, it deformed the tissue, and yeah, it took forever.
But these researchers built a resource they call LAMBADA, which stands for Light Sheet Aligned Mouse Brain Annotated Developmental Atlas.
Right. And the way it allows them to see through solid tissue is by combining cutting-edge technologies. The first is a tissue clearing protocol called iDisco+A.
Okay, how does that work?
So the reason brain tissue is opaque is mostly because of fat. Lipids scatter light. So through a very precise series of chemical baths using solvents, they literally strip the fat out of the brain tissue.
They just dissolve the fat.
Exactly. And they replace it with a liquid that allows light to pass straight through without bending. The brain becomes completely transparent, like glass.
Oh, wow.
Yeah, and then they use fluorescent antibodies that bind specifically to blood vessels. When they shine a laser through this clear brain using a light sheet microscope, all the surrounding tissue is invisible.
And only the glowing vascular network shows up in perfect 3D.
Exactly.
So they basically turn the brain invisible, highlight the plumbing with neon paint, and take a 3D picture. That gives us the physical structure. But they didn't stop there, because just seeing the pipes doesn't tell you who's ordering the plumbing.
Right. Structure alone only tells you where the roads are. To understand why they're being built, you need to know what the cells are actively saying to each other.
So they integrated a second technology, right?
They did. It's called Visium. This is a spatial transcriptomics tool. Normally, if you want to read the RNA like the genetic instructions a cell is sending out, you have to grind up the tissue.
Which means you lose the map of where those instructions came from.
Exactly. It's a smoothie. But Visium allows them to take an ultra-thin slice of the brain and read the genetic instructions while keeping the exact spatial coordinates of where those instructions were issued.
I want to make sure I'm visualizing this right for the listeners. It sounds like having a flawless 3D Google Earth map of an entire city's road network. That's the iDisco plus clearing.
Spot on.
But then you add a real-time filter over that map that lets you read the text messages of all the construction workers building those roads exactly where they are standing. That's the Visium tool.
That is a perfect way to visualize it. You see the road and you simultaneously read the cellular text message saying, hey, we need more asphalt at this exact intersection.
That is wild. You get the anatomy and the biology at the exact same time.
Yeah. You do. But merging those two things created a massive computational headache. When you image a massive 3D network of tiny capillaries, the software trying to trace the lines often makes mistakes.
Like glitches in the matrix.
Exactly. It might see a sharp turn or a dim spot in a vessel and accidentally render it as a broken line or a dead end.
Right, a glitch in the Google Earth map. But in biology, a dead end in a blood vessel is actually a real thing, isn't it?
It's a very important thing. It's called a tip cell. A tip cell is the literal growing front of a new vessel. It has little microscopic fingers that push through the brain tissue to form a new connection.
Wait, so the software just sees a broken pipe, but the biology says, no, that's a living cell trying to grow.
Exactly. So the researchers had a massive problem. How do you separate the software glitches from the real biological tip cells that are actively building the network?
You can't exactly have a grad student click through millions of microscopic dead ends to verify which is which.
No, you really can't. The data set is too immense. So they trained an artificial intelligence, specifically a machine learning model called SWIN-3D.
SWIN-3D.
Yeah. It's a model originally designed to help AI understand the 3D layout of indoor rooms, but they adapted it for biology. They fed the AI thousands of verified examples of true biological tip cells.
And thousands of examples of mere software glitches, I assume.
You got it. The AI learned the subtle physical characteristics of a growing vessel versus an artifact. It independently cleaned up the adult brain maps by a staggering 91%.
That is a huge leap. The AI wasn't just enhancing an image. It was effectively acting as a biological proofreader. It had to learn the physical signature of a living cell.
It's incredible technology.
Okay, so let's do a quick recap for you listening. We've got this new tool, LAMBADA. It uses iDisco Plus to make the brain transparent, Visium to read the genetic text messages of the cells, and a SWIN-3D AI to clean up the glitches.
That's the foundation.
So think about a map you use every day on your phone. How much more useful would it be if it told you not just where things are, but why they're being built there? That's what LAMBADA does for the brain.
Exactly. And using this master map, the researchers looked at the first few days in a mouse's life, from postnatal day 3 to day 7.
Okay, phase 1.
Right. And what they observed is something they call isometric expansion.
Isometric expansion. Let's break down what that actually looks like. Iso meaning equal, metric meaning measure.
Exactly. During these first few days after birth, the brain is growing rapidly in sheer physical volume. It is ballooning in size, and the total length of the blood vessels is also increasing incredibly fast.
So everything is just getting bigger.
Yes, but here's the fascinating detail. The density of the vessels stays exactly the same across the entire brain.
Imagine you take an uninflated balloon.
Yeah.
And you take a marker and draw a grid on it, like a lattice of equally sized squares.
Okay, I'm with you.
Now, you blow the balloon up, the balloon gets bigger, the lines you drew stretch out and get longer, but the number of squares stays exactly the same. The grid is just expanding uniformly.
That's a great analogy. The blood vessels are essentially just stretching out to keep up with the rapidly growing brain tissue. It's a very uniform, non-specialized growth.
So the brain isn't playing favorites with any specific region yet?
Not at all. But if we look at the Visium data, those genetic text messages, we can see why this is happening. What is the actual driver pushing this growth?
Right, because something has to be giving the order.
And it turns out this initial phase is driven primarily by a state of hypoxia.
Hypoxia, meaning low oxygen.
Yes. Because the brain tissue is expanding in volume so incredibly fast, the cells are literally getting pushed too far away from their existing blood supply.
Oh, because there's a physical limit to how far oxygen can travel.
Exactly. There's a limit to how far it can diffuse through tissue. When the cells get pushed beyond that limit, they experience mild suffocation. They gasp for air.
That sounds intense.
It triggers a very ancient canonical survival response. They release a specific protein called VEGF-A, vascular endothelial growth factor A.
So VEGF-A is basically a microscopic distress flare. The tissue is screaming, "We are suffocating, send oxygen!"
Precisely. And that VEGF-A signal tells the existing blood vessels to just blindly sprout new branches. It's a biological copy-paste mechanism.
There's no complex blueprint yet.
None. The genetic driver is purely structural survival.
Okay, so to sum up phase 1 for you guys, from day 3 to day 7, the brain is undergoing isometric growth, like our balloons stretching uniformly, and it's driven entirely by hypoxia and that VEGF-A distress flare.
Just trying to keep the lights on.
Exactly. So have you ever experienced a time where you were just stretching to keep up with rapid changes rather than specializing? That's your brain in phase 1.
Beautifully put.
But clearly a brain cannot just expand evenly forever. Eventually it has to start processing complex information.
And that transition brings us to phase 2. This runs from day 7 to day 21. And this is where the landscape at the brain completely changes.
What happens?
The sheer volumetric expansion of the tissue starts to slow down. But the vascular density, the amount of plumbing packed into a specific area, suddenly skyrockets, and it doesn't happen evenly anymore.
The brain splits into 2 distinct tracks,
Yeah.
right?
You have high-density regions like the hindbrain, the thalamus, and the cerebellum, which suddenly become incredibly thick with new blood vessels.
And the low-density ones.
Regions like the striatum and the cortex, they develop their networks much more slowly. And the timing here is critical. This day 7 to day 21 window is exactly when the baby mouse starts truly experiencing the world.
Right, the sensory boom.
Exactly. It opens its eyes, it starts actively whisking its surroundings, the sensory floodgates open, and the neuronal computing power required by those specific relay stations goes through the roof.
Now, hold on a second. If we're saying sensory input drives the blood vessels, does that mean the exact second the mouse opens its eyes, the visual cortex gets flooded with new vessels?
You know, that is an incredibly logical question. And honestly, it's exactly what the researchers assumed they would find. They thought the visual cortex would spike the moment the eyes opened.
But it doesn't.
No. The surprising reality is the data showed something completely unexpected. The sensory regions don't align their vascular growth to their own specific neuronal critical periods.
Wait, so the visual cortex doesn't act independently when the eyes open?
No, it aligns with its parent region's overall dynamics. It just follows the broader schedule of the entire cortex.
That is so weird. It's like, okay, think of it like a city planner who says, we are upgrading the entire downtown grid this year, rather than just laying new pipes for one specific building the day a new tenant moves in.
Yes. It's coordinated on a larger macro scale.
That makes a lot of sense from an efficiency standpoint. But if the trigger is shifting from just gasping for air in phase 1 to processing the world in phase 2, the genetic text messages must be completely different.
Oh, they are. They completely shift.
The cells can't just be using that VEGF-A distress flare anymore.
They aren't. That hypoxia signal basically fades away entirely. To figure out what replaces it, the researchers used a complex mathematical algorithm called CNMF to group thousands of different active genes into specific biological programs.
And what did they find?
They found that the genes driving vascular growth in phase 2 are intimately linked to neuronal activity. We see immediate early genes, which are essentially the genes that fire the moment a neuron sparks with electricity.
Wow.
We also see massive genetic signatures for synaptic maturation.
So the neurons are actively calculating their own power needs. They are forming new synapses, firing off electricity, and directly calling for blood supply based on their computing demand.
Exactly. The infrastructure is actively adapting to the behavior of the brain, and the blood vessels actually anticipate this demand.
How do they anticipate it?
Well, the researchers tracked a specific type of endothelial cell, the cells that make up the blood vessels, that express a protein called ERG. Think of the ERG protein like a biological ID badge worn only by mature, specialized construction workers. In phase 2, they saw these ERG-positive cells actively migrating and redistributing themselves ahead of the actual vessel branching.
So the specialized workers are literally showing up to the site before the road is even scheduled to be built.
Yeah.
That is incredible predictive infrastructure.
It really is.
Okay, let's recap phase 2 really quick. From day 7 to 21, the uniform expansion stops. Hypoxia fades, and the brain starts packing in dense blood vessels based on regional schedules driven by the active firing of neurons.
And we get that divergence into high and low density regions.
Exactly. So what stands out to you listening right now about the fact that your brain's physical infrastructure was literally shaped by your first experiences of the world?
It profoundly reframes early childhood development, doesn't it?
It really does. The literal plumbing inside your skull wasn't just a generic blueprint. It was custom poured by the first times you opened your eyes and heard a sound.
It's amazing. But it also introduces a massive biological risk.
Uh-oh. What risk?
If the blood vessels just keep growing endlessly every single time a neuron fires, the brain would become a chaotic, tangled mess of hypervascularization. It would flood the system. It would be entirely unsustainable.
Right. If you just keep laying asphalt over every inch of the city, nobody can actually live there. At some point, you have to put up traffic lights and pour concrete to lock the grid in place.
You need biological brakes.
Does the brain have biological concrete?
It absolutely does. And that brings us to phase 3, stabilization and the brakes. This phase runs from day 21 well into adulthood.
Okay, so what happens in phase 3?
During this phase, the overall density of the vessels finally stabilizes, the rapid sprouting stops, and the focus shifts entirely to refinement. For example, they looked at a marker called SM22.
What does SM22 do?
It measures the maturity of the thick, muscular arterial walls. You need strong, mature pipes to handle the high pressure of a fully functioning brain.
So they're reinforcing the main supply lines.
Exactly. And they found that in phase 3, the brain locks into a highly consistent mathematical rule. For every one arterial branch, there are exactly 100 capillary branches.
Exactly 100 to 1.
Yes. It doesn't matter what region of the brain you are in, that 100 to 1 ratio becomes the golden rule of the stabilized network.
But how does it actually stop the growth to maintain that ratio? You mentioned biological brakes. What is physically stopping those tip cells from pushing forward?
There are two main actors pressing the brake pedal here. First, there is a repulsive guidance molecule called SLIT2.
SLIT2, what does that do?
It acts like a biochemical do not enter sign. Wherever SLIT2 expression is high in the brain tissue, vascular density drops.
How does it actually stop the cell, though?
When an actively growing blood vessel hits a gradient of SLIT2, the physical cytoskeleton inside the endothelial cell collapses. It literally loses the structural integrity to move forward, repelling the vessel away.
Wow, so it chemically strips the vessel of its ability to walk forward. That is a hard break.
It is a very hard break. But the second break is perhaps even more structural, and it involves astrocytes.
Astrocytes, those are the starch-shaped support cells, right?
Yes, exactly. Early in development, immature astrocytes actually permit or even encourage blood vessels to grow. But in phase 3, as they mature, they flip a functional switch. They start expressing a marker called AG, or angiotensinogen.
So the support cells change their behavior. How does AG stop the growth?
When they mature and express AG, the astrocytes physically change shape. They extend these structures called end feet and wrap them tightly around the blood vessels.
Like a microscopic corset.
That's exactly it. They form a corset around the capillaries, which is a key part of the blood-brain barrier. By physically clamping down and tightening their grip, they prevent new branches from breaking through the wall. They transition from permissive builders to strict physical regulators.
That is the biological concrete setting. The astrocytes mature, wrap their end feet like a corset around the pipes, and the grid is locked.
It's locked in.
Now, there was one detail in the source material about this locked-in phase 3 that absolutely fascinated me, and it has to do with biological sex. We tend to think of male and female brain development as something that happens very early on in the womb.
Right, the sexual dimorphism of the brain.
Exactly. But the researchers found that differences in the blood vessel networks between male and female mice do not appear in phase 1 or phase 2.
That's true. During those phases, the plumbing looks identical.
The structural differences only emerge in phase 3, which correlates with puberty and adolescence.
That's right. Around day 30, when the hormones kick in, they found significantly higher vascular density in specific regions of the male brain, like the corpus callosum and the tuberal nucleus.
And the female brains.
Female brains showed higher density in entirely different regions, like the diagonal band nucleus. It perfectly illustrates how phase 3 isn't just a static shutdown.
It's still refining.
Exactly. It's a period of highly specific, hormonally driven refinement where the brain is making its final long-term structural adjustments.
Okay, let's do a final bullet recap for phase 3. Arterial walls mature with SM22. The brain enforces a 100 to 1 capillary to artery ratio using brakes like the SLIT2 repellent and ACT from mature astrocytes. And this is when sex differences finally emerge.
You've got it perfectly.
So knowing that biological brakes like mature astrocytes lock your brain's vessels into place, how does that change your perspective on how permanent our early developmental phases are?
It's a profound question.
If I am putting this all together,
Right.
We started with the immense challenge of wiring a supercomputer while it's actively running. We've seen how the brain solves this through three masterful phases.
Right.
First, a uniform survival-based expansion driven by cells gasping for oxygen. Second, a highly specialized regional densification where active neurons summon their own power supply based on sensory experience.
And
finally, a locked-in astrocyte-stabilized support system that enforces order with chemical repellents and microscopic corsets.
it is a profound shift from a state of raw survival to a state of tailored, experience-driven efficiency to ultimate regulation. But as we wrap up, I want to leave you with a thought that builds on all of this, something to mull over.
Oh, lay it on us.
We know that adult learning and memory require the formation of new synapses. If you learn a new language or master a complex physical skill, you are building new neuronal connections in your brain.
Right.
And we now know, thanks to this LAMBADA atlas, that synaptic maturation is the exact trigger that drives blood vessel remodeling in phase 2.
Okay, I see where you're going with this.
So the provocative question is this. Even though the mature astrocytes have applied the biological brakes, does our adult brain still quietly unlock these hidden developmental mechanisms to reroute microscopic blood flow every single time we learn something new?
Oh, wow.
Are we, in tiny, invisible ways, constantly loosening the corset and re-entering phase 2 just to feed a new idea?
That is a brilliant thought. Is my brain physically rerouting its plumbing right now just from processing the concepts in this paper? It really makes you realize that your brain isn't just a static computer sitting in your skull. It is a living, breathing, constantly shifting landscape.
It's a beautifully dynamic system.
And that is exactly the kind of question that keeps us doing this show. Thank you so much to everyone listening for joining us on this deep dive. Keep questioning the incredible machinery inside your own head, and we will see you next time.